I'm Charlotte Mackay, Head of Trade and Investment at the British Office Taipei. I'm delighted to welcome you to this year's BIOAsia Taiwan 2022 exhibition. In this UK Regional Collaboration Forum, I'm pleased to announce that 9 highly sophisticated UK companies, both new and long-standing organisations, will be presenting today on their exciting and innovative products and areas of expertise. We will also present an overview of the UK life science environment and what we've achieved as global leaders in the life science sector. In sequence of streaming order, you'll find introductions from leading UK biopharma firm AstraZeneca, anti-tremor pioneer GyroGear, automated cell cloning platform IOTA Sciences, state-of-the-art microscope provider Oxford Instruments and/or, genomics expert Yourgene Health, advanced 3D printing company AMT, best-in-class biotech consultant Evaluate, UK biopharma and science leader GSK, and radiation therapy innovator Leo Cancer Care. I hope you'll find the information on display insightful, and I would be delighted to invite you to visit our UK pavilion to talk directly to the UK company representatives here and also to the staff of the British Office Taipei. Come and learn the potential for collaboration with your business. Please enjoy the video and enjoy learning about the mysteries of human science.
我们都想跟爱的人在一起。陪伴他们的时间总是不够。再多倾听都听不腻，都是心意。为他们奋战，是你理解生命的意义。你拼命，因为想要有更好的生命。你努力，希望好的事降临。最好的事就是和爱的人好好的在一起。因为你知道，每次离家出门，最后真正的目的地都是家。保护好爱的人，保护好自己，这是你的坚持。坚持好的事，坚持对家人好的事。坚持好好的在一起。AZ 坚持做对的事。
Dr. Salia Hassan has been to meet an inventor who is helping people for whom that sadly isn't true.
Much of what we do without thinking relies on precise coordination of our hands. Something that for many, like Alison in Bath and Tom in Essex, can be extremely difficult.
Say that plug there, I can't even put that on there because I shake about too much.
OK.
It's just ridiculous.
Like over 1 million people across the UK, Tom and Alison suffer with a severe hand tremor.
It's just a challenge using normal things like a dustpan and brush. When I go to hold it, the dust just flies everywhere.
People don't realise how disabling it is. I'd give anything to stop doing this so I could live a bit more normal like.
Tremors are caused by neurological problems like Parkinson's disease in Alison's case, or essential tremor in Tom's. And unfortunately, medication doesn't always work. So Tom and Alison are taking part in a new trial that hopes to transform the lives of sufferers.
Good afternoon.
I'm Salia.
How are you?
Hi.
Come on
Good,
in. Come on in.
Thank you.
Inventor Fai Ong was a medical student when he decided to try and tackle the problem. Not through medication, but with physics. He decided to build a device based on a very simple technology.
What we really thought was exceedingly elegant as a solution was a simple gyroscope.
A gyroscope is a gadget that's designed to remain steady and in the same position. As the wheel inside the mechanism spins, a force is generated, keeping the gyroscope upright. Fai believes the same concept could stabilise hand tremors.
So our basic understanding is that if we take this and fasten it to the hand, then we have at least a good chance of reducing tremors as best as possible.
Fai has incorporated industrial-strength gyroscopes into a prototype glove that he's trialling with sufferers.
All right, welcome to our little office.
Right,
Let me just grab a seat here, if that's all right.
Tom's come in for his very first test session.
Jubbly.
The glove is still in the development stage.
Let's wait for it to get up to speed.
Today, Fai is assessing whether the fit and size of the device is appropriate for Tom's tremor. Fai's aim is to make a glove that is small, quiet and portable enough to enable users to perform everyday tasks. While the team continue to tweak Tom's glove, Alison has come in for her second test to see whether her customised glove will work, and she's desperate to find something that can help her.
So it's 6 years I've been diagnosed, and it just does get worse.
To show me her struggles, Alison tries to perform one of Fi's regular tests, to draw a simple spiral.
Just can't get anywhere near, and you know what it's supposed to look like.
Yeah.
The frustration. It's the frustration, yeah.
With the glove already adapted to Alison's hand tremor, will it make a difference?
Oh my God. That is like, I'm so blown away by what I've just
tearful.
seen.
I find it hard because it's controlling your hand again.
Wow.
It's the most amazing sensation to have your hand back.
How do you feel?
It's just wonderful because you feel like you've got 2 hands again.
It's a remarkable result for Alison and a great step for Fi and his team towards releasing their first glove to target those who need it most.
Being here today has been a real privilege. Watching Alison draw that spiral and that look on her face when she did it was priceless. It was so emotional.
And if the trials work out, it could help transform the lives of thousands of people who suffer from this debilitating condition.
Isn't that fantastic?
Some of the stuff we're hearing and seeing on tonight's programme. Thank you, Salia. We've spoken to Alison and she's heading back to the lab this week for more tests, and they do hope to perfect the glove early next year.
Good news.
Sue was diagnosed with a disorder called essential tremor 5 years ago, and now finds even the most basic tasks a challenge. But newly designed special gloves are helping people with Sue's condition and Parkinson's disease regain control of their lives.
To me, it's a big thing not being able to put a bit of makeup on and not to be able to put a necklace on and change my earrings. If I want to change my earrings, I can't do it unless there's somebody here to help me. I just can't get them in and out. I can't paint my nails anymore. My daughter does them for me. I can't cut my nails. I can't cut my toenails. There's all sorts of things that I can't do, but these are going to enable me to do a bit more, keep more of my independence instead of losing it day by day.
As long as it's not too tight around your...
No, it's fine.
The gloves have built-in spinning gyroscopes. Mounted on the back of the hand, it counters any tremor, smoothing out the shakes. Developer Fi Ong was inspired to tackle hand tremors while caring for an elderly woman in hospital who was struggling to feed herself soup.
So we have an aerospace-grade gyroscope in this. It functions exactly like a top. So as the top spins, it tries to stay upright. So in the same way with this, the glove actually counteracts your hand tremors immediately and instantaneously. So as you shake, it automatically cancels out the shakes.
Ong puts his medical career on hold to develop the product. He hopes it'll be given medical approval in the US and EU next year. And for sufferers like Sue, they say it's the helping hand they need.
Hi, I'm Anna, and I'm a scientific officer from IOTA Sciences, a biotech company from Oxford. Here, I would like to introduce our new automated cell culture platform that addresses critical bottlenecks in traditional single-cell cloning methods. Single-cell cloning is involved in diverse applications. It is a process of obtaining a homogeneous cell population, be it by their genotype, phenotype, function, or morphology. However, the only way that guarantees that a population of cell is homogeneous is by clonal expansion from a single progenitor cell, and this process remains challenging. Traditional single-cell cloning approaches like FACS sorting may be too harsh to cells, leading to reduced survival of already fragile cells because they are in a single-cell state. Single-cell cloning via limiting dilution and manual colony picking also has its drawbacks, as they are labor-intensive and may use expensive reagents or consumables. These methods are also time-consuming, not only because of picking and pipetting, but because one has no upfront assurance of monoclonality before cell expansion. For example, when cloning via limited dilution in a 96-well plate, it is almost impossible to see a single cell in the well. Due to optical edge effects, it is also difficult to spot a colony itself, which is hidden here. After waiting for cells to expand into colonies, we assume that a densely packed colony comes from a single progenitor. Unfortunately, this is often not the case, as exemplified here, where a round colony is not pure. To overcome these challenges, IOTA Sciences' cloning platform utilizes small-scale cell culture chambers with unique optics that allow the identification of single cells directly after plating. Here are images in a 4x objective showing the whole chamber, where a single cell is clearly identifiable even if it's located at the edge of the chamber. 256 individual chambers form a grid, and this grid is made entirely of fluids, your cell culture media, and an immiscible transparent fluorocarbon. Each chamber utilizes less than 1 microliter of media. This here on the right shows a clonal population of adherent cells, and at the bottom, of suspension cells after a week in culture. IOTA Sciences' single-cell cloning platform is automated by the IsoCell and the IsoHub. Let me walk you through our simple workflow. The IsoCell fabricates the grid chambers and automatically dispenses cells into the grids. Identification of chambers containing single cell is done with the help of the IsoHub that features a microscope and works as an information and control hub. Throughout the entire workflow, the IsoCell and the IsoHub automatically synchronizes data wirelessly with each other. This allows users to be able to easily decide on which chambers to feed, track, and harvest into PCR tubes or 96-well compatible strips for further expansion and characterization. IOTA Sciences' cloning platform is highly efficient in cloning adherent and suspension cells. In addition, our system is compatible with commonly used coatings and is shown to produce high cloning efficiencies in iPSCs. Please visit our virtual booth or our website for further information. Do contact us if you have any questions on how we can improve your single-cell cloning. Thank you.
Hello, I'm Geraint Wild from Andor Technology, and I'm here today to tell you about the BC43, a brand new novel benchtop microscope to boost your productivity. So, life exists in 3 dimensions, but often people image in 2 dimensions. Why is that? It's predominantly because most accessible and affordable microscopes are only really capable of imaging in 2 dimensions, or if they can image in 3 dimensions, it's only to a limited depth. But of course, many samples are quite thick, and if you want to see your protein of interest in its true context, you really do need to see it more often than not in 3D. So here's an example. With a conventional wide-field epifluorescence microscope, looking at, say, this solefish head, which is 521 microns thick, if you're trying to look at the nerve cells in cyan or the muscle cells in yellow, it's really hard to see any detail. You can see some detail, but you have all of this hazy out of focus information. If you were then to move into confocal imaging, then all of a sudden, all of that detail becomes that much clearer. So now you can see nerve cells very clearly through the spine here, and then you can see them coming out and innervating the head of the soulfish and even the muscles here that you can see in yellow. So confocal imaging has a true benefit when you're trying to image anything in depth. Not only that, when people are doing confocal imaging, it's not just on fixed samples. They want to be able to look, for example, at dynamic processes in cells, see how their proteins behave in dynamic processes, but then also in multicellular organisms here, like the Drosophila egg chamber, right through to entire tissue sections or whole animal models, for example, like zebrafish and Drosophila. And not only that, we want to see them, say, in living samples as well as fixed. So there's these multiple scales that need to be covered, and also the ability to do both live sample imaging as well as fixed sample imaging. So when we were designing our BC43, we were keeping all of this in mind so that you get the most benefit out of a single instrument. So how did we make confocal imaging more accessible? So this is what a traditional confocal microscope could look like. They're large, they're complex, and they do indeed carry out very sophisticated experiments. But many people who use confocals actually only want to do relatively routine imaging on them. So it seems more efficient to produce a confocal microscope that's more readily usable for routine imaging, leaving these more expensive, complex instruments available for those who want to do more complex experiments. And these would typically reside in a specialized core facility with somebody who could, an expert who can manage and help you use these, and in dark rooms, for example. So they're not so accessible to an everyday research or research group, I should say, working in their lab. So we basically took the technology that's required for routine imaging in a confocal microscope and condensed it down to this, the BC43. So here you can see this benchtop microscope with a lid so that you don't need a dark room, and it sits very happily on a benchtop alongside devices like microcentrifuges or mini vortexes. It takes up very little space at all and is now in your lab and can be part of a complete workflow where you're preparing your samples and imaging them at the same time. So what is in a BC43 microscope? It's actually a multimodal imaging system. So it has at its heart a multi-point spinning disk confocal, but it is also capable of wide field fluorescence microscopy. There are times when that can be of benefit. And also it's capable of imaging unstained or non-labeled samples using transmitted light microscopy. And here we have a bright field, which is a standard regular imaging methodology. And then also we have something called differential phase contrast, which allows you to see some contrast in samples that would otherwise have no contrast and gives us sort of a 3D relief image, which I can show you an example of later. So what's a multi-point spinning disk confocal? Well, basically, inside the BC43 is a disk with thousands of pinholes, and that spins at high speed. And those pinholes basically ensure that you only capture in-focus information and not out-of-focus information. So that when you're imaging through a sample, and when you're imaging through a fixed sample, what you're doing is imaging one focal plane at a time and stepping through the depth of the sample and then reconstructing an entire 3D image. And so that disk with pinholes gets you the most in-focus image. And because it's a disk spinning at high speed, it captures in a single snap with a camera, unlike more traditional confocals where they scan one line at a time and it can take seconds to capture a single image. So also on the BC43, we have real-time 3D visualization. We're able to show you in real time your image in 3D developing as each individual section is captured. This is great because if you've set up your experiment parameters wrong and you need to start again, you can identify that very quickly, stop, and then reset your parameters. A lot of solutions out there, you'd have to wait to the end before you can actually see that there's any error in your 3D volume. We have 4 laser lines, 405, 488, 561, and 637. So these will excite your most commonly used probes like DAPI, FITSI, GFP, TRITSI, DS-RED, and the likes of Alexa 640. Have a unique illumination source called Borealis. This gives us maximum uniformity across the whole sample, which is great for accurate analysis from edge to edge of your image. But also if you're doing large imaging very large specimens like that intestine ring that I showed you earlier, it means that you have to do multiple images to capture such a large specimen at high resolution, and then stitch those images together. And when they're stitched, you can't see the join. It's perfectly flat. The BC43, as you can see, doesn't have any eyepieces. So we have something called Focus Seek, which helps you find your sample in the first place. You can find your sample in seconds. And then also the same mechanism can be used to lock on to your sample when you're doing time-lapse experiments, so that, for example, if the temperature in your laboratory changes, the sample will always stay in focus throughout the whole time series. We have a technique called deconvolution, clear view deconvolution, which I'll come to. And also, most importantly, it has an anti-vibration mechanism built into it. So in doing so means that you can use it on a bench that has devices that can cause vibration, like mini centrifuges and vortexes. So I mentioned that BC43 is a high productivity microscope. So here's an example. This is a Drosophila egg chamber. It's 93 microns thick, and we have basically two proteins labeled here, one in cyan and one in yellow. And basically we've captured two colors, and we've taken a volume over 309 optical slices. So that's 309 slices through the sample. And we've done this at full resolution. So that's a full field of 2048 by 2000 pixels. So in total, we've captured 618 images, and we've captured those in just 2 minutes. A traditional point scanner would take you close to an hour to capture the similar data set. So that's how much more productive a confocal the BC43 is. Here's another example. So this is somebody, a researcher who has been looking at intestine in zebrafish. And in this case, they want a high resolution image so they can zoom in and see the detail, but they want to be able to capture the whole tissue section that they've got. So in order to do this, they've captured a number of images, neighboring images, and then stitched them all together. So, and this is also a 3D image. So in total, they've captured 77 Z planes through their sample. There's 4 colors here, so 4 different fluorophores. And then this whole image is made up of 28 individual images that are stitched together. So in total, that's 8,624 images, and that's been captured in just 30 minutes. Again, if you were to use a more traditional point scanner, that would take you 6 hours or more, depending on your experimental settings. So I mentioned that BC43 is capable of live cell imaging. So here on the left, actually, and on the right, are 2 examples. On the left is wide field version, the wide field modality of BC43, in combination with the differential phase contrast I mentioned. So you can see that even in unstained cells, you're able to see the cells. And this is in wide field on the left. And then on the right-hand side, we have the same experiment, or similar experiment, I should say, in confocal. Only in this case, for productivity, during the time lapse, we've captured multiple positions at each time point. So here you've got 4 fields being captured at the same time. So for example, if you were wanting to analyze, the more fields you can capture, the more cells you can analyze, and therefore the better the statistics of your experiment. And what's important to note that we have got full cell division here. Confocal microscopy can often cause phototoxicity, and phototoxicity will stop cells from dividing. So here it's clear that the BC43 is not phototoxic because we can see cells happily dividing. I also mentioned earlier that we have a technique called deconvolution. We have with Andor, we have clear view deconvolution. Other names for deconvolution might be image restoration or digital clearing. This is a methodology that basically, particularly for those people who are maybe using wide field microscopy, this algorithm basically helps take away the haze. Typically the haze is out of focus information from somewhere else. So what the image restoration or deconvolution does is it's an algorithm that knows where that light came from, and it reassigns it back to its original place. So on the right hand side here, this was captured in wide field, for example. And if we use view deconvolution, you can see how much clearer that image becomes. So all of that information is restored back to where it came from, and so you have a higher resolution, higher clarity image. And actually, even though convolution is excellent at giving a high clarity image to start with, in some cases deconvolution can improve that image even further. And of course, you want a highly detailed, high contrast image, because if you then want to visualize that, for example, here using 3D surface rendering, or for downstream analysis, for example, counting cells or counting structures within cells, you need to have the best quality image before you start to do that kind of analysis. With Andor, we have the Amaris 3D visualization and analysis engine. It's the market leader. And so we can take these images that are now really crisp and contrasty and again, use them downstream for analysis. And Amaris has many, many packages for cell biologists, cancer researchers, neuroscientists, all with tools specially designed for some of the typical analysis that you would do in those research fields. So how can BC43 help you? Well, if you're a core facility manager, then you have a product now that is ideal for routine imaging and giving you high quality images, confocal images, but it does mean now that you can free up those more expensive, sophisticated confocals for the experiments that they're designed for, that people want to use them for. So it helps reduce the queue, if you like, for those more sophisticated instruments. There's minimal training and therefore also minimal user support, so it gives you more time to manage your facility. It's great for inexperienced users, so you don't have to worry about them damaging the system or getting lost in imaging settings. It's low maintenance, so it's got low running costs, and it's highly productive, so you can serve more users with it. And then in addition to those advantages, if you're in an individual research lab, for example, then the ability to put the BC43 at the heart of your lab increases your productivity, puts it in the same space as your sample preparation. Now you're able to move from 2D to having actually a high quality fluorescence 3D microscope in your lab, saves you having to go and access a core facility. And it's really easy to learn. So you don't have to be a microscopy expert to use this BC43 confocal microscope. So I'd just like to thank you for your time and for listening to me. I hope you can see the benefit that the BC43 could bring to your research lab and how it could accelerate your research in the future. Thank you.
Welcome everybody, I'm Stephen Toe, the CEO of Leo Cancer Care, and I'm here to talk to you today about innovations in upright radiation therapy. When we founded Leo Cancer Care, we founded it to be the more human way to deliver radiation therapy. And this sounds like a very abstract idea, but for the Leo Cancer Care team, it's very, very clear exactly what that means. We are driving to improve the clinical effectiveness of radiation therapy through the use of upright patient positioning. We're improving global access to radiation therapy by replacing the need for expensive rotating gantries with fixed radiation and very slow patient rotation. And finally, we're empowering patients and clinicians to face radiation therapy together, eye to eye. So let's look at those in a little bit more detail. Radiation therapy, as it is today in the conventional sense, operates around two core principles. There are many devices out in the market today that look in some ways very different, but they all operate around the principle of radiation therapy being delivered in the supine or prone orientation, with the patient lay down. They also all operate around the idea of radiation that rotates around the patient, and radiation that can weigh anything from 6 tons all the way up to 600 tons. Until now, Leo Cancer Care is changing both of those two core principles. And it's really an innovation of two parts. First, we've developed an upright patient positioning system capable of treating everything from the head and neck down to the prostate. All of those key indications like head and neck, breast, lung, and the prostate. And partnered with that upright positioning system, we've developed an upright dual energy CT, capable of imaging all of those key indications. So how does that work? How does upright radiation therapy work? Well, it really starts with patient posture optimization. So in a conventional sense, when we treat patients supine or prone, we tend to treat head and neck or breast or lung or prostate in exactly the same position, with the patient lay down on the table. In an upright sense, we're able to optimize patient posture based on the indication. So for head and neck patients, we treat in a conventional seated position with the shoulders dropping naturally by the side. For breast or for lung, we can lean the patient slightly forward to bring the breast away from the chest wall. And for prostate patients, we can position in this semi-standing or perched position. And in this perched position, you completely open up the pelvis, allowing for anterior beams in through the front of the patient. And what's incredibly important here when moving into this approach is to be able to position patients with limited mobility in this semi-standing position. And for that, we use a combination of a shin rest to support the mass of the patient across their shins, and also a heel rest to come and support the patient behind the feet to stop them from sliding back. So once we have the patient in their optimized posture, we're able to bring down a diagnostic quality dual energy CT at the isocenter. We're imaging the patient to ensure that the tumor is in the correct location. And once we've verified that, that's really the easy step. We can then bring in the radiation, and that can be photons, protons, carbon, helium, or even neutrons. The effect is exactly the same. And the final step, again, moving away from this idea of large gantry rotation, is to very, very slowly rotate the patient in front of that fixed radiation beam, delivering beams from different angles in exactly the same way as we would with conventional radiation therapy. So I talked right at the start about that driving improvements in clinical efficacy. What do I mean there, and for what indications specifically? Well, some of the biggest centers all over the world, places like MD Anderson and Northwestern, MGH, and the Paul Shearer Institute, have all published for these 4 key indications, stating the fact that the organs move less in the upright orientation. And with a reduction in organ motion, it should drive us to be able to deliver radiation more effectively. And just across these 4 indications, where the literature already suggests this today, that already accounts for 66% of all cancer incidences. So let's just take a look at one of those in more detail. MD Anderson, and in particular Lawrence Court's group down at MD Anderson, published a paper a number of years ago looking at lung volume in the upright orientation compared to supine. What you're seeing here on screen is the same patient, the same phase of the breathing cycle. The only difference is that the image on the right is in the upright orientation. And we can see a 50% increase in the volume of lung in that orientation. So for the same tidal volume of air, what we see is that increase in lung volume results in a reduction in breathing motion. And the paper again from MD Anderson showed a 4 to 5 millimeter reduction in lung motion. So for a tumor, for example, that was in the lung, you would see that same reduction in tumor motion, and therefore allowing us to treat that tumor more accurately. So that's one very tangible example of the clinical advantage, or the anatomical advantage, to treating patients in the upright position. But there's also a psychological benefit. We as humans feel more in control when we're in the upright orientation. It's a position of power. Being supine, if I was presenting to you today supine, it would just be a little bit weird. It's not a position where we connect and it's not a position where we feel in control. So we believe that treating patients in the upright orientation will give that better connection and more empowered feeling for the patient. Let's just have a quick look now at the numbers. I mentioned that this was improving global access to radiation therapy, and it's doing that by reducing the size, the cost, and the complexity associated with radiation therapy. This is a snapshot of protons, and you can see in the center of the screen a typical rotating gantry for proton therapy that you might see today. The radiation in the bottom left-hand corner, the bit that produces the protons that treat the patient. And you can see that the equipment in the center here is incredibly large. This is in the order of 110 tons and requires a 3-story building to rotate the radiation around the patient. And it almost always actually makes sense to rotate the lighter object. If something has to rotate, it's much easier to rotate the lighter object. And that's exactly what Leo Cancer Care is doing. So rather than taking the radiation, feeding it into this very large structure and rotating it around the patient, we keep the radiation fixed and slowly rotate the patient instead. This saves in the order of around about $20 million overall for a project involving fixed beams rather than gantries. A complete game changer in the health economics of proton therapy. And it's not just equipment cost or building costs. When we look at total cost of ownership, we find it easier to install these devices, to maintain them, to power them. All of these things that come into the total cost of ownership reduced if we move away from the idea of rotating the radiation and instead simply rotate the patient. And just a fine example here of the project at UCLH in London, which shows just the magnitude of building projects when we consider proton therapy in a conventional sense. Leo Cancer Care is talking about taking this kind of construction site that we're looking at now and putting that into a conventional linear accelerator vault. Total game changer in the footprint of these type of therapies. So in summary, upright patient positioning reduces motion during treatment, meaning radiation can be delivered more precisely in that upright orientation. Utilizing fixed radiation beams rather than rotating gantries dramatically reduces the size, the cost, and complexity associated with radiation therapy. And finally, being upright is just a more human posture. Leo Cancer Care allows patients and clinicians to connect eye to eye during their therapy. Thank you very much for listening.
Hi everyone, my name is Giorgio Ioannidis and I'm Global Head of Sales for AMT. Thank you very much for the opportunity to do this presentation. Yeah, we're going to present our company, give an overview of AMT and also our products, and at the end, a quick summary of what we spoke about and I guess the key message, take-home message. So who is AMT? We're the world leaders in automated post-processing. What does that mean? So essentially, we're a hardware manufacturer, so we make machines for the 3D printing industry. And the type of machines are called post-processing machines, so the finishing machines of 3D printed parts. We were founded in 2017 by Joseph Crabtree. We've had exponential growth. I actually joined the company in January 2018, so just over 4.5 years. And from that time to now, we've grown the company to about 120 employees across three sites, including Hungary, the UK, and the US. We're forecasted to turn over 60 million by 2024. We've sold quite a few systems to date, so over 200 systems in about 28 countries. And we've also created global partnerships with leading 3D printing OEMs, including HP, 3D Systems, And then we work with other companies like Saint-Gobain, which are actually one of our investors. So the leaders in abrasive and surface solutions. Another one of our investors is DSM, the chemicals company, the Foresight Williams Group, and so on. We like to say that we've smoothed or finished a lot of parts, and obviously this is quite a hard number to estimate, but we've estimated around 3 million parts have gone through our technology, and that number is growing every single day. We've also generated a lot of our own IP, so we have over 40 patents which have either been granted or are patent pending, or waiting for the grant to go through. So yeah, we have achieved quite a lot in the last few years. A bit about our facilities. So we are a global company. We have our own manufacturing facility in Veszprém, Hungary. So that's where the design, the R&D, the manufacturing actually happens. So the biggest workforce of our company is in Veszprém in Hungary. It's about an hour away from Budapest. We have our own facility in Austin, Texas, and that's mainly to support the North American market. This is more for customer support, customer experience, and so on. And then where it all started was in Sheffield in the UK. And again, that's a customer experience center, the fundamental R&D, and the customer support for the rest of Europe. We don't have a presence in APAC just yet, but we do have 2 people there, and they're supporting the machines that we have over there from sales and support. So our capabilities are actually quite different, I would say, to our competitors, and it's actually a competitive advantage that we have. We design, develop, and manufacture the machines in-house. So we have full control of the process, which means we can bring new products to market quite quickly. And if there's a need for a custom machine or a custom solution, we can actually do that. So we have full control of that workflow, and we don't subcontract manufacturing for our vapor smoothing systems. We fully do that by ourselves. And a lot of the software that is also developed in-house. And that comes on to a later discussion about the automation and robotics when I speak about our DMS system. So really, what's our mission and vision? We're trying to provide an automated finishing technology for every 3D printer. So right now we're focusing more on thermoplastic polymers. However, there's many different types of printing technologies out there. It could be metals, different types of powders for metals, extrusion processes, ceramics, resins, and so on. So there's many different types of printing technologies, and we want to have an automated solution for all of them, and obviously mainly focusing on the industrial applications. The vision of the company is to enable 3D printing as a manufacturing technology. So right now, a lot of the benefits from 3D printing are actually eroded because the post-processing is so expensive and it takes so much time. And I'm going to dig into that into the next few slides. So once the part has been printed, it actually needs to go through a number of steps before it is a finished component. For this presentation, we're mainly going to focus on powder bed fusion, and that tends to be our biggest client base. So that could be an SLS printer, a multi-jet fusion printer, a high-speed sintering printer, and so on. So anything plastic powder is what we really focus on. So for a 3D printed part to perform like its traditionally manufactured counterparts or injection molded part, it needs to have an improved surface finish, or it needs to be cleaned or depowdered. It needs to be coated or painted or dyed, and so on. And then at the end of it, of course, inspected. So all these post-processing steps are very manual. And obviously, when you have a manual process, it's slow, it's expensive, and you can't keep up with the throughput. So if, for example, you have 50,000 parts to print and finish, then when it comes to finishing, if everything is manual, then it's extremely labor intensive, it's slow, the costs go up, so you end up falling into a lot of issues, and there's no real solution unless you automate the workflow. So this is just an example of an HP multi-jet fusion part. It's actually, that one is a TPU part, but it's the same for other polyamides and so on. When the build has finished, this is what it will look like. So this is not what other OEMs advertise. They advertise nice and shiny, smooth finished parts, but that's not the truth, really. So they have when they come out of the printer, they're covered in powder, they have a porous surface, they're rough. The properties maybe aren't ideal for the application. There's no color, so there's a lot of things that need to happen to that part to make it a finished product. And I'm going to go through the stages in the next slide. So currently, what happens is you have your build cake. Once the print is finished, it needs to cool down. It needs to be unpacked, which essentially means you need to separate the bulk of the powder from your 3D printed components, which have been sintered. Again, that's pretty manual. There's a limited number of automated machines out there, so most companies will be doing this in a manual way. Once the parts have been unpacked, which they will look like the second picture, then they need to be cleaned or fine cleaned, which means complete removal of any unsintered powder. Which the way it's done now, it's manually bead blasted. So you've got air pressure, you've got some sort of media. It could be glass beads, it could be poly beads, ceramic beads, a lot of different types of media. Anyway, some sort of media along with blasting air pressure, and then you're cleaning the part. Super manual, labor intensive, and needs to be automated. And then finally, the vapor smoothing, which could be essential depending on the application, of course, whether it's a consumer product or a medical product or an automotive product. In many cases, it will require some sort of surface finish because the nature of the 3D printing process, you have these layer by layer, so they call it the stair-step effect, which causes a surface roughness on the part. Again, manual processes could be literally a manual immersion or a coating. Again, super expensive and time consuming, so that's another step that needs to be automated. So what do? We offer automated machines or different modules to deal with this problem. So as I said, the first stage is unpacking, then you go to the fine cleaning, depowdering. You could also do a surface blasting, which essentially means to lower the surface roughness of your part with an abrasive media. And then the final surface finishing, which is the vapor smoothing to make an injection molded like finish. So we provide all these machines, and I'm going to go through a few of our products and the solutions that we currently have. The unpacking is something that we're still working on, but it's going to be launched very, very soon. Commercially, we have depowdering, surface blasting, and also the chemical vapor smoothing. So the first commercial machine that we have is the fine cleaning. This is done in a machine, or we have different machines which are very, very similar. They have a cyclone within them. It's a fully ATEX and CE certified system within an enclosed environment. So when you're dealing with powders, you want it to be enclosed in a system because one, it causes a mess in the facility, and two, it's actually a health hazard. So just from a safety and health perspective, it needs to be done in a machine to avoid the operator being in contact with any sort of powders. So the systems that we're offering, starting from small and scaling up, depending on the type of printer and production, size of production, we have the DP Studio, DP, DP Pro, and DP Max. So they all these systems work in a very, very similar way. Essentially, you're using blasting at high pressure air, 4 or 5 bar, let's say, along with an abrasive media, could be glass beads, poly beads, and so on, to clean your parts or free them of powder. Obviously, the DP Studio would be suited more to a desktop style machine, let's say. And then as you go up, DP, DP Pro, DP Max, you're looking at more industrial solutions, predominantly DP Max. So the DP Max could handle multiple industrial 3D printers, whether it's an EOS P396 or a 3D Systems Pro X or an HP Multijet Fusion 4200 or 5200 system. So multiples of these systems the DP Max can handle. The DP Max is a bit more different to the other three because it has a belt system. So it's a rotating belt where the parts would physically loaded in. And again, the reason is because you can fit more parts to be cleaned in one go. And it's a continuous workflow. Whereas the DP Pro, DP, and DP Studio have a rotating basket. So a tumbler, a basket where you put the parts in and they're tumbled around while being blasted with media and air pressure. So that's a bit of the depowdering. Actually, one thing I missed was a quick comparison of manual versus automated. So if you put your parts in these machines or a batch of parts, let's say a build from an industrial printer, you can probably finish that in about 10 to 15 minutes. If you were to manually blast these parts, so let's say you had 300-400 parts to manually do, that could be a number of hours, right? It could be 2 to 4 hours of manual cleaning. So a significant reduction in labor and time. Whereas here in an automated system, you place your parts, you press a button and the process finishes. Now onto the surface blasting. For blasting, essentially you can use different machines or the SB surface blasting machines to improve the surface roughness of the parts. And essentially it's, again, a very similar process to the depowdering, so I won't go too much into that one. So the vapor smoothing is our core product, and that's the one that we design, develop, and manufacture internally. And this is years of research. We took a lab scale process and turned it into an industrial solution. We have two types or sizes of vapor smoothing machines: the SF50 and SF100. SF stands for surface finish, and the number stands for the liters of the chamber, but both machines have the same footprint and virtually work the same way. A quick comparison to our Post-Pro 3D, which was our first generation machine. We've managed to get a 75% reduction in size, moving from Post-Pro 3D to the SF machine. So the SF machines are the ones that we currently sell, both the 50 and the 100. And they're about a fridge size, let's say, so that gives you an idea of how big the system is. So actually quite small, that takes up small space in the facility floor or the factory floor. The SF100 Plus is something more recent that we've been working on, and that's some software updates to improve the throughput and essentially turn over or churn parts much quicker. And also which gets you a reduction in the cost per part. So what we're doing with the vapor smoothing, we're actually going from 10 to 12 microns, let's say, on a powder bed printer, all the way down to about 1 to 2 microns, depending on the material. We're also sealing the surface. We're improving the mechanical properties, predominantly elongation at break, which significantly goes up. The surface roughness, of course, as I said, goes all the way down. If the parts were colored, it becomes brighter, so more apparent colors. And also the parts can be sterilized and cleaned very easily, or wiped clean, versus the porous as printed parts. We can deal with a lot of different types of materials, from powder bed fusion or extrusion. And as I said, we focus right now on thermoplastic polymers. So that could be the polyamides, the composites, so carbon-filled nylon, glass-filled nylon, the elastomers like TPUs, the PBAs, TPEs, and so on. So we can handle a lot of different types of thermoplastic materials. Now, quickly just finishing off and moving to the DMS, the digital manufacturing system is really what the vision and where we're trying to get to as a company. And these are custom solutions that we're working on already. And basically, it's taking all these automatic modules of different types of machines, putting them together, and creating an automated workflow. So automatic loading and unloading with robots, inspecting, scanning systems, automatic racking, bin picking, so picking and placing parts and sorting them, and so on. And this is basically a lights out manufacturing process. There is a video here that I will share in a link because I'm running out of time. So I will share that separately, which explains a bit more about the DMS. So summarizing what I've just spoke about and about AMT, as we said, we're a company that are automating the post-processing workflow. And without this automation, the benefits of 3D printing are eroded. So it's the only way that really we can have a viable manufacturing process via 3D printing for high volume or scaling production. We're the patent owners for the chemical vapor smoothing, and that's our core product. And that's our flagship product, which we're pushing out to the market. And we've sold over 200 of these systems globally and continue to sell these week by week. We have real life applications and real life production enabled with AMT Post Pro. We actually have a number of case studies that I can share. Again, both in medical, automotive, and various other applications that without the chemical vapor smoothing, those parts would not be able to be used in production or for the end use application. So yeah, sorry that I've gone on for more than 50 minutes, but I'll try to keep this as short as possible. This is some contact information from our commercial team. Feel free to contact me directly at giorgio@amtechnologies.com with any questions. And of course, Damson, who is our market director for the APAC region, would probably be a good contact to have with any questions. He'll be more than happy to support you with any of your questions. So once again, thank you very much for your time. Thank you for listening. And yeah, look forward to hearing from anyone very soon. Take care. Bye-bye.
As a biopharma company, consultancy or investor, how do you identify, validate and prioritize opportunities, maximizing the value of your asset, portfolio and, ultimately, your organization? Often, what's needed is to bring in independent expertise for trusted validation on internal models or hypotheses, or specialist assistance with uniquely sophisticated analysis. Evaluate Consulting and Analytics are here to help you solve your specific biopharma pipeline, portfolio, and commercialization challenges. We support you with best-in-class datasets, powerful analytical capabilities, and deep expertise from both a commercialization and therapy area perspective. Our defining difference? We can draw on the whole of Evaluate's data and analytical capabilities, along with our team's broad expertise, and our primary and secondary market research capabilities to support you across a wide range of requirements. When you need to identify and assess new business opportunities, reduce risk, raise the quality and speed of decision-making, and obtain strategic expert recommendations, make us your port of call. Evaluate Consulting and Analytics. Definitive answers to your unique and complex questions.
Chen Beidar is Evaluate's Chief Consulting Officer, and he works closely with pharma organizations and investment companies to help them make the best decisions for their companies. He's joined today by Amy Brown, Deputy Editor of our Evaluate Vantage service. She's a journalist with extensive experience in biopharma and the financial markets, and many of you will recognize Amy's name from the many articles and reports that she's written for Evaluate.
So this first slide is a look at what will be the biggest sellers this year. Obviously, we're dominated by COVID still. Consensus forecasts for the antivirals was only just coming in when we wrote the preview 4 months ago. Since then, billions of sales have been added for this year alone. I think this is all going to be a flaw for these COVID numbers. I think they're only going to go one way, and that is up. There's a lot of contracts signed already. Obviously, as we all also already know, the pandemic remains very unpredictable. So we will have to see how those numbers develop over the year. Outside of COVID, this is going to be the last big year for Humira, AbbVie's big anti-TNF. It's hard to overestimate how massive this product has been. It's been the best-selling drug since 2012. That took over from Lipitor. It's hard to think that we're going to see another product that could quite amass the sort of lifetime sales we've seen with Humira, if only because of the kind of political efforts to curb such kind of monopolies. So this next view is a kind of another way of looking at where the sector is spending all its money. We've got, this is an Evaluate Omnium analysis. They do, they take real world data and company disclosed R&D spend to throw out these estimated costs of clinical programs. These are an estimate, I would stress. This is you'll always get the projects that have got the big cardiovascular outcome studies, the big long-running trials that will rank highly here. The final section for me is just a quick look at the regulatory environment. This is just the FDA's novel drug approvals. I mean, I think there was a lot of concern during the pandemic that we would see the regulatory process almost grind to a halt. I mean, that obviously didn't happen. There were certainly delays, and perhaps the last couple of years would have been even bigger if we hadn't have had those. But I mean, I think historically, the last couple of years don't really look too weak. This year, I don't think any massive changes expected. You know, the FDA's finally got a permanent commissioner, Robert Califf. I think he's viewed as a fairly safe pair of hands. But it is the FDA user fee program reauthorization this year. I think PDUFA 7 we're on to now, possibly PDUFA 8.
Hello, everyone. So when we look at the stock market, we see that biotech has been in a bear market since early last year, and the current global environment hasn't helped to make it more attractive, unfortunately. While we don't know where the bottom is, we know that this impacts the short-term valuations and momentum to access public markets. That's where it brings us to the IPO market, actually. For the IPOs, unsurprisingly, we don't see a lot of activity within IPOs. We have just seen one file this week with AN2 Therapeutics recently. However, we feel that there are companies waiting in line until the market conditions get better. Although this seems a bit depressing, the fundamentals are very strong with the R&D productivity, proving that the emerging mechanisms of action and technologies are getting better at solving unmet needs. As we have seen with mRNA during COVID. That's why we see a confidence in private investments, such as venture capital firms, actually. And when we look at VC and private investing, if you take out the peak in Q1 last year, the trend is actually still strong. However, we have started seeing the late stage ventures converging to public market valuation. Early stage companies are still priced at premium, and this will probably continue a bit like this until we see a lot more decrease in public market valuation, impacting both the early stage and the late stage biotech company firms. And if you look at the biopharma buyouts, understandably, Q1 has been a bit quiet, given the fact that what's going on in the global landscape. There have been announcements around from Pfizer, UCB, and Viatris, but we have yet to see some significant momentum. The premiums are still high, especially at the early stage, as I mentioned. In light of these facts, probably one question that we could ask is that whether we will be able to see premiums going down a bit and we start seeing more of the M&A activity. And I think we have to wait and see to test this hypothesis. I mean, there's generally no correlation between big pharma and M&A buyouts, but certainly we don't have a crystal ball right now, but probably we need to wait and see what the market will bring us in the near future. Finally, when we look at licensing, which is probably one of the best signals, which shows confidence in innovation R&D, is we see the upfront licensing fees are still on the rise, cementing this confidence. Although we haven't seen a blockbuster deal yet, given the fact that the public valuations are coming to a more chewable state, we believe there will be opportunities, as I mentioned before, in the coming months. And actually, by looking at the projects that we have been involved in, I can say that there have been some significant activity boiling up recently. both in terms of licensing opportunities or searching for licensing opportunities, or in terms of small to mid-sized acquisitions. As a summary, we can confidently say that pharma has strong fundamentals with the R&D machine working smoothly at the moment. Yeah, we see some kind of failures in the late phase stages of the clinical development, but there's still the machine is working really efficiently and smoothly in terms of bringing up or testing new or emerging technologies to the marketplace. I guess also there has been some kind of a recent article from Morgan Stanley touching on this as well, looking at the pharma productivity and R&D. In that regard, we have 4 recommendations to pharma companies. First of all, you should keep continuing investing in R&D programs, especially in new technologies and unmet needs. For example, and again, I mean, I've given this example before, but messenger RNA has shown its potential during COVID. From the partnership perspective, it's crucial to identify and establish partnership with early stage companies, not from the fact that building a portfolio, but also from the fact that to orchestrate the drug discovery ecosystem, which will be very impactful in 5 to 10 years. However, when you are investing in early stage companies, what we also recommend is the fact that, especially with the emerging technologies, people should take a look at it from a different perspective, shifting from assets or portfolio of assets, or managing the portfolio of assets, to shifting it to managing the risks involved in the clinical development programs, or adopting a risk budgeting portfolio management scheme going forward. Finally, digital technologies. I guess no one could deny the impact of digital technologies. I mean, we have seen a lot of impact, improvement coming out of market. There have been several IPOs specifically in this space, both leveraging AI and ML to support drug discovery. We see a huge increase in the number of applications of digital health or digital therapeutics to develop and integrating novel digital biomarkers in diagnosis, monitoring, and managing diseases. So with that said, it's going to be an integral part of the pharma or the life sciences to leverage digital technologies in your existing portfolio of products.
有劲基因是一家结合技术开发和服务供应的企业，运用在基因医学相关领域上。我们与国际知名大厂合作，把开发的检测产品成功地推广到60个以上的国家。为了提升企业营运效能，有劲今年成立了分子生物卓越中心，坐落于远雄 UTown 商办大楼，并配置了最先进的科技与技术，提供客户良好的教育训练环境，以此扩展有劲基因与当地企业、大学及医院的合作。
有劲基因成立10多年来，一直致力于基因定序的服务与相关的应用开发。高规格、高精密的次世代定序实验室是我们能够提供高品质服务的关键。实验室拥有良好的动线规划，实施人员与简体的动线分流，空间规划上进行分区，设置有负压区、缓冲区，也有设置即时环境监控系统、紧急电源等设施，让简体的风险降低到最小。毕竟每一个实验简体都是无比珍贵的。次世代定序技术是推动精准医疗发展的关键。一个医药级的次世代定序实验室，加上一群经验丰富的人才，让有劲基因能够成为学研单位、医疗单位、药厂或生技公司的关键服务伙伴。 无论在基础的基因体学研究、研发、临床试验，或是基因检测上，都能够扮演重要的推手。
台湾有机基因位处于亚太地区重要的位置，恰好能够服务东北亚及东南亚的客户。除了检测服务之外，我们也提供技术转移服务，将我们开发出来的检测服务项目能够复制到其他国家。此外，我们也提供检测试剂及分子生物学实验仪器的销售与技术支援，及时的解决亚太地区各个经销商还有技转客户的技术问题。台湾的行销业务与技术支援团队已经成功将产品及服务销售到亚太地区各个国家。
GSK started more than 300 years ago in 1715 and has grown into one of the largest pharma, vaccine, and consumer organizations in the world. 2022 is a monumental year for the organization. We will separate our consumer business and create two world-leading businesses, one in consumer and one in biopharm. Our biopharm organization will remain under the name of GSK. Our purpose is to unite science, technology, and talent to get ahead of disease together. Our R&D is focused on the science of the immune system, human genetics, and advanced technologies, investing in 4 therapeutic areas and future opportunities to bring impact health at scale. We operate responsibly for our stakeholders by prioritizing innovation, performance, and trust. Our culture has 3 pillars. 1, ambition for patient, to deliver what matters better and faster. Accountable for impact, with the ownership and support needed to succeed. All underpinned by doing the right thing with integrity and care, because people count on us. GSK Taiwan was established 42 years ago, and today we have over 300 employees. Our general medicines portfolio offers treatments covering over 50 different medicines, highlighted by a comprehensive respiratory, CNS, and urology portfolio. Our vaccine portfolio has over 15 vaccines licensed in Taiwan. Nearly every newborn every year is receiving our pediatric vaccine or vaccines. In our healthy aging segment, we have a wide range of vaccines to help protect adults as they age so they can remain healthy and strong. This year alone, we've launched 2 important vaccines, one in the pediatric space and one in our healthy aging space. We will continue to bring innovation to Taiwan to support Taiwan to be healthy and stronger in the future. Lastly, our HIV business offers over 10 medicines to treat people living with HIV in Taiwan. We will continue to launch new innovative HIV medicines to improve the quality of life of those individuals. We have 3 ambitions in Taiwan. One is we want to expand the impact of our value to more patients and individuals in Taiwan. Today, more than 2 million Taiwanese see the value of our vaccines, our medicines each year. We want to expand that impact. We want to do that with our vaccines and our medicines. 3 examples of that we're doing right now is expanding and strengthening our trusted vaccine portfolio by launching new assets. And we're going to continue to do that. We also want to make sure that we are a vital partner to CDC and the federal government tenders. We're going to continue to do that. It's important from a public health perspective. For HIV, we want to empower the future people living with HIV. We're going to do this with new innovative launches, which we've started in 2015 and already launched 3 new medicines. And we're going to continue to do that over the next 3 or 4 years. For our current portfolio that is treating people with chronic conditions, we want to continue to support them with those medications. And we want to bring in new treatments for people that are living with lupus, CKD anemia, severe asthma, and continue to bring innovation to Taiwan where there is an unmet medical need and we can have an impact. Second is to value diversity and inclusion. Simply, it's the right thing to do. And it makes any organization better when they really see the value and value the inclusion and diversity of an organization. We will ensure every individual at GSK, regardless of background, regardless of gender, regardless of sexual orientation, that they have an equal footing to be successful and have a voice at GSK. Today, our organization has 58% of our managers and my LT are female. We're going to continue to value that. As June just closed, we still know that there is a stigma within Taiwan and frankly across the world with the LGBTQ community. GSK is an ally. I am an ally. And we're going to continue to support important events in Taiwan like the Pride Parade and BCCT Diversity Awards. Because at the end of the day, love is love. And being able to be your true self can only bring the best out of you. Lastly, we're going to be a caring and compassionate organization. We want to do that for our employees and the communities that we live in. For the communities that we live in, every year, every employee is supported to take a day off to give back to their community through local charities or national charities. We call this Orange Day. Last year, even during the pandemic partial lockdown, we were able to do these programs through virtual ways. For our employees, we want to provide monthly afternoons that they can take off and just recharge. We call these Awesome Fridays. This just enables people to just step away from work and recharge and come back, be able to be more productive because they have the proper life balance. Secondly, we offer free vaccines to our employees and their family members for any vaccine that is recommended by WHO. We are going to continue to look into the right projects, resources to support as the world changes. So we're at this place right now where Taiwan is going to be super aged. How can we support people as their parents age as an employer? We want to make sure that we do that. Lastly, we need to evolve how we work, right? Through COVID, we figured out that working virtually or at home in the office is totally fine. And I think we want to continue to allow that and for employees to make that decision where they want to work, when they want to work, and provide them the resources to be able to do that. We call this performance with choice, and we'll continue to have that as we go into the future. All these ambitions is really about supporting everyone at GSK to get ahead together. Thank you.